Phylogenetic trees provide a graphical representation of an evolutionary history of a group of organisms, with tree branches representing populations through time. The evolutionary history of a group of organisms is called its phylogeny. A phylogenetic tree is a graphical summary of history, showing the ancestor-descendant relationships among population, species, or higher taxa, and clarifying who is related to whom. The tree of life is the most universal of all phylogenetic trees, depicting the evolutionary relationships among all living organisms on Earth. The root is the most ancestral branch represented in the tree, and a branch represents a population through time. Any horizontal line is a branch. A node represents a specific hypothetical ancestor, in many cases the most recent common ancestor of two separate branches. A tip, or terminal node, represents the end point of a branch. An ancestral trait is a characteristic that existed in an ancestor, and a derived trait is one that is a modified form of the ancestral trait found in a descendant. A synapomorphy is a trait found in two or more taxa that is present in their most recent common ancestor, but is missing in more distant ancestors. Synapomorphies allow biologists to recognize monophyletic groups, also referred to as clades or lineages. A monophyletic group or clade includes an ancestral population and all of its descendants, but no others. A monophyletic group includes an ancestor and all of its descendants, but no others. Monophyletic groups are distinguished from polyphyletic groups and paraphyletic groups. A polyphyletic group is an artificial grouping that does not include common ancestors. The branches shown in purple are an example. A paraphyletic group includes an ancestral population, but does not include all of the descendants. The historical relationships among species cannot be known with absolute certainty. So the relationships depicted in phylogenetic trees must be estimated from the best available data. The most commonly used method of inferring phylogenetic trees, called cladistics, was introduced by German biologist Willy Hennig in the 1960s. The cladistic approach is based on the principle that relationships among species can be reconstructed by identifying shared derived characters, synapomorphies. Although it is not simple in practice. Sometimes the species form a monophyletic group one way, according to one trait in the matrix, and a different way according to a different trait in the matrix. The larger the number of species and traits added to the matrix, the greater the number of possible trees and character combinations. There are 15 possible trees for a phylogeny of 4 taxa, but over 100,000 trees for a phylogeny of 8 taxa. How can researchers identify the tree with the best fit to the data? How can they avoid an erroneous conclusion? Biologists often invoke the logical principle of parsimony to address these questions. Under parsimony, the most likely explanation or pattern is the one that implies the least amount of change. Parsimony analysis uses computer programs that compare the branching patterns that are theoretically possible in a phylogenetic tree, and they count the number of changes in DNA sequences required to produce each pattern. For example, the tree in figure B requires only 2 changes in base sequence, while the tree in figure C requires 4. Since independent evolution of traits should be rare compared to, say, similarity due to shared descent, the tree that implies the fewest overall evolutionary changes is hypothesized to be the one that most accurately reflects what really happened during evolution. Trees created using cladistic analysis are called cladograms, and they focus on the branching patterns. Some other types of analyses produce trees whose branch lengths represent genetic distance or time since divergence. You can identify these trees by the presence of scale bars. The cladistic approach is a powerful method of estimating phylogenies because it is based on one of the central concepts in evolutionary biology, homology, meaning same source. Homology occurs when traits are similar due to shared ancestry. In contrast, homoplasy, meaning same form, occurs when traits are similar for reasons other than common ancestry. A common cause of homoplasy is convergent evolution. Convergent evolution occurs when natural selection favors similar ways of making a living in different species. The extinct aquatic reptiles called ichthyosaurs were strikingly similar to modern dolphins. Both are large marine predators with streamlined bodies and large dorsal fins. Are these characteristics homologous in dolphins and ichthyosaurs? The figure on the right shows a phylogenetic tree based on skeletal characters, such as the number and placement of openings in the skull. Since ichthyosaurs are known only from their fossilized skeletons, this analysis shows that dolphins occur within the mammal clade, whereas ichthyosaurs are most closely related to lizards. Notice that the sister groups to dolphins and ichthyosaurs, that is, the most closely related branches on the tree, do not possess streamlined bodies, flippers, and fins. Dolphins and ichthyosaurs look similar but are not closely related. Based on these data, it is logical to argue that the similarities between ichthyosaurs and dolphins result from convergent evolution. In homoplasy, the convergent traits do not occur in the common ancestor of the similar species. Streamlined bodies, flippers, and dorsal fins are adaptations that help any aquatic species, whether it is a reptile or a mammal. It helps them to move quickly in open water, hunting or fleeing.